The Friends of Farm Foundation program can help you further expand your network with a diverse group of food and agriculture leaders. Entry-level benefits include virtual networking sessions, while contributions at the upper levels unlock additional benefits, such as a personalized tour of our new Innovation and Education campus. Help us celebrate 90 years of making a positive impact on the future of agriculture. Visit farmfoundation.org to explore the benefits of becoming a friend and supporter of Farm Foundation today.
Good afternoon. Thank you for joining us today for our Farm Foundation Forum, Innovation in Gene Editing and Plant Breeding, a look at scientific advancement and consumer perspectives in food and agriculture. We're glad to have the opportunity to engage with you and are thankful to Farm Credit for their support of the forum. My name is Sherry Rogie-Fiddler, and I'm the president and CEO of Farm Foundation, located just outside of Chicago, Illinois. I'm looking forward to today's discussion, which will look at the scientific advancements happening in plant breeding and gene editing in food and agriculture, as well as the impacts for agricultural productivity. Beyond the science, we'll hear updates on the regulatory environment, as well as insights into consumer perceptions, preferences, and demand. Before we get into today's forum, I'm going to take just a couple of minutes to share a bit more about Farm Foundation. We are a 501c3 nonprofit working at the intersection of agriculture and society to address challenges that affect the entire food and ag value chain. Specifically, we're an accelerator of practical solutions for agriculture, accelerating people and ideas into action. This year, we are celebrating 90 years of doing this work. The three levers we use to accomplish this are policy, innovation, and education. Forums such as today's are just one part of an extensive program of work, which is guided by our mission to build trust and understanding at the intersection of agriculture and society, and our vision to build a future for farmers, our communities, and our world. We rely on partnerships to fund our work and increase our impact. So if you're interested in learning more about funding or partnering with us, I invite you to reach out to explore collaboration. Now I'd like to take a minute to highlight our Friends of Farm Foundation program. With your enrollment as a friend, you will not only be helping to support the mission and vision of Farm Foundation, but you will also gain exclusive benefits such as first reads of our issue reports, networking opportunities, and much more. Being a friend is an investment in building a better future for farmers, our communities, and our world. To learn more about being a friend of Farm Foundation, go to farmfoundation.org/friends. The link is also being posted in the chat. In addition to learning more about Farm Foundation and our work by visiting our website, I encourage you to connect with us on our social media platforms. If you're posting on social media about today's session, we ask that you please use #FarmFoundationForum. And here are the last few housekeeping notes. There will be an audience question and answer session at the end today. We'll be using the Q&A button at the bottom of your screen, not the chat, to queue up the questions. This forum is being recorded and will be posted on our website at farmfoundation.org, as well as our YouTube channel. We'll send out that link following today's program. And when the forum concludes today, you will receive a link to a brief survey. We really appreciate your feedback and your time in completing this survey. So now let's turn to today's forum topic, innovation in gene editing and plant breeding. Today's outstanding panel will be able to provide a diversity perspective on this important topic. It is now my pleasure to introduce, as our moderator and discussion leader, Dr. Jayson Lusk. He is currently the Vice President and Dean of the Division of Agricultural Sciences and Natural Resources at Oklahoma State University. From 2017 to 2023, he served as Distinguished Professor and Head of the Agricultural Economics Department at Purdue University. He is a food and agricultural economist who studies what we eat and why we eat it, and is a prolific researcher, writer, and speaker. He's also a Farm Foundation Roundtable Fellow. We're so pleased to have you as our moderator today, Jayson, and at this point, I'll hand the baton over to you.
Thanks, Sherry, for those kind words. It's a pleasure to be here and a real honor to be able to get to moderate this session. So I thought I'd make a few remarks before turning it over to our main speakers. I've had the good fortune of spending 20 plus years studying consumer demand, consumer reactions to food and agricultural related biotechnology. and market responses. And if I had to summarize a lot of that work very succinctly, I would say that most people in most places know very little about food and agricultural biotechnology. So as a result of that, what we see is how you frame questions, what labels you put on products, what framing you put around the topic, what information you give can have substantial impacts on whether people find food and ag biotechnologies acceptable or unacceptable, worth eating, not worth eating, or what premiums they might be willing to pay to either have or avoid some of those technologies. And so I think because of that, I think it's really, useful and important to understand what benefits technology can convey in biotechnology. I think the research also suggests that the less foreign, so to speak, an intervention might be in terms of biotechnology, the more acceptable it is to the public. So because of some of those things, I've long thought that the answer to consumer acceptance of food and ag related biotechnologies might rely in the technology itself. Meaning, can technology evolve in ways that create food and agricultural products that people can relate to, that provide more direct benefits for them, that occur in ways that seem less foreign. And so I think that's one of the reasons I'm really excited about this panel, is to hear from our panelists about what's happening on the science side of things. And I look forward to hearing what they all have to say. So without further ado, I'm going to introduce our first panelist, Dr. Alan Van Dynes. He's the director of the Seed Biotechnology Center and associate director of the Plant Breeding Center at University of California, Davis. His research focuses on developing and integrating genomics into plant breeding of California and African crops. He has programs on breeding for disease resistance and quality in pepper and spinach, and development of applications of genomics in crops. He's co-developed and is organizer for the university's Plant Breeding Academy and is past chair of the U.S. Plant Breeding Coordinating Committee. So thanks for joining us, Dr. Allen. I look forward to hearing what you have to say.
Thanks, Jason. And I think that's my son's picture up there. It's a pretty old one. Next slide, please. Thank you for inviting me. And today, I want to kind of echo a little bit of what Jason just said on the consumer side with one of our collaborators who's speaking today, Dr. Finley Chou. I wrote an article about a year and a half on the importance of gene editing for sustainability and nutritional security worldwide in the United States. And it went out to hundreds of thousands of subscribers to, very large servers and things like that worldwide. And I thought, okay, I'm going to get all sorts of emails and hate mail and things like this. And I only got 2 responses, 2 emails, and both of them said, "Thank you for writing that." And so I think it just really echoes what Jason said. This is not the main thing people are worried about today. It certainly wasn't, the response I thought. So I'm going to jump into gene editing, and what we've been working with. And the title says an opportunity to play. And part of that is, unlike GMOs, for most part, small companies, small crops, and public has been kind of out of the game. And we hope this is something that we can, really use and get out and get products out. Next slide. And to give you an idea of where gene editing might fit, this is something I put together with my colleague, Dr. Rita Mam, a while back. We have, gene editing is just one of the tools that plant breeders use, and there's a continuum of new tools that are developed. And if you go back to the original tools of really understanding genetic principles with Gregory Mendel back in 1865 when he first released them, and we go on to learning about statistics, experimental design. I put biotechnology. Notice it's kind of in the middle there. Really, in the 50s, late 50s, we talked about tissue culture, mutation breeding. Transgenics came around in the early 80s. And genome editing in the early 2000s. But we have all sorts of other technologies that we're working with, like speed to market technologies, counter-seasonal nurseries, This is what Norman Borlaug and the Green Revolution really counted on. But machine learning, genomics, high throughput phenotyping, artificial intelligence, and then, of course, intellectual property and education. So again, it's gene editing is just something that plant breeders and animal breeders and so on, will use. Next slide. So where does gene editing fit in? First of all, I'd like to define plant breeding. So plant breeding is really a discipline of sciences. It's not just one science, but it's rooted in population genetics, selection theory, quantitative genetics, and statistics, but also has a whole number of increasing support technologies like the previous slide I mentioned, just to name a few. Next slide, or next slide again. So GMOs are as the traditional transgenic technologies, were really new sources of genetic diversity in the breeding cycle. Whereas gene editing is also a source of genetic diversity, where plant breeders create new genetic diversity. They select the best lines, or really take out the undesirable lines, fix those traits, release varieties, and then continue to improve with those. Gene editing gives us an opportunity, though, if we can apply it really into our elite materials. And that way we can really just tweak or improve something that is already, pretty much very good, except maybe missing one trait or two traits that we like to fix. So we can bring gene editing closer to the end of the breeding cycle. And I want to point out that in any breeding program or any breeding cycle, we test over generations, we test over multiple field locations, and years and so on. And I think this is critical for everybody to remember that, people don't just create things in the lab and put them out, and hope for the best. Next slide. So since I'm the first speaker, I'm going to quickly define what gene editing is. First, I want to talk about, traditional GMOs, or gene modifications. That is inserting a gene, whether that's from the same species or another species, into the plant, or animal, and getting, a phenotype from that. The challenge with that is the technology is actually quite difficult. It's limited to the number of hosts and the number of varieties or genotypes that we can do. It requires plant regeneration, and it's actually quite low efficiency. Give you an example, most products have had to have at least 100 to even 1,000 different insertions of genes before they found one that they really liked and worked well. Say in canola or in corn, those were some of the first gene edits in soybean. Next slide. Sorry, gene modification. Whereas gene editing is just like it says, you don't necessarily need to integrate any foreign DNA into the genome. You actually use things like CRISPR-Cas9, which is a group. It's a protein or a group of enzymes that bacteria use to defend themselves from virus, and we sort of hijack it, and we can say, well, maybe we can get this enzyme to only change a gene that is already there, as opposed to insert a whole new one. And so that's a different concept, I think, in consumers' minds. And what Cas9 does, it cuts the DNA in double-stranded DNA, and it turns in where you can actually just rebind those two pieces, or you can actually insert a separate piece where that cut is. And it turns out that DNA repair system in plants and animals and humans is not perfect. And when you cut that DNA, sometimes you'll get a piece of the DNA missing or a small insertion. And so that's how it works. So basically, we're tweaking a gene that is already there, or we're inserting something at a very specific locus in the chromosome. Next slide. Gene editing is not one thing. People know about CRISPR. There are many, many different CRISPRs. There's about 13 or 14 now, different versions of it right now that do different things. Some insert things, some just change one base to the other. Some work on the promoters. Next slide. And one of the things we can do with gene editing, but something we can do through regular plant breeding, is what we call gene stacking. So that's just putting a bunch of genes together. Plant breeders have been doing gene stacking for a long, long time. This is a publication, an old genetic map from Dr. Gerdif Kush, who won the World Food Prize in 1996 for all his work in rice. And he's a colleague. His office is next door to mine, who works at an alumni at UC Davis. But where he just created the first maps for genes, and he found that some of the genes were close together, they were linked, and he was able to move those genes together as one piece. And so a long time ago already, back in 2000 and when was this? 2008, the Collier et al., this is a group out of USDA in Albany, tested if they could use gene editing technology or GMO technology combined to add genes one at a time and continue to add up to 25 genes all in the same place in the genome. Well, this is a breeder's dream, where all those genes, let's say these 25 genes, one could be disease, one could be drought tolerance, yield, so on, quality, flavor, they all are inherited as one block or one unit. So the breeding is much, much simpler. Next slide. So what are our breeding goals in 20 years? They're not that different than they were 20 years ago, but where I think we have better tools to do things with much better efficiency. I think the urgency is getting larger, because we don't only have increasing population, we have climate change, we have environment, and that's also changing which pests, which challenges we have. We can increase nitrogen use efficiency, nitrogen fixation in crops, flavor, quality, and so on. And I want to say, even at UC Davis, we have projects in every one of these, with every one of these goals working. And some of them are real. I'll give you the next slide. Gene editing can be used, and GMOs can be used for breeding tools. So doubled haploids are a type of tool that we can use, traditionally done through tissue culture, but you can do it as an inducer slide, where you make a cross with a specific parent that has a modification in it that basically allows it only to replicate, allows it to eliminate its own genome. So you basically only get, for example, the maternal genome is expressed. You can double that with a compound called colchicine, and now you have a pure breeding line in one generation. And this is some technology that came out of UC Davis and is actually being applied, I know, in many crops right now with industry. Next slide. Hit the next slide one more time. This is something that came out last September, or 2 Septembers ago, from my colleague, Dr. Eduardo Blumwald, just down the hall. Using gene editing by simply knocking out 2 genes in rice, he was able to basically re-engineer nitrogen fixation in rice. And this has been a target for many, many years. I know people have spent literally $500 million trying to make nodules that legumes have, put them in corn or another cereal grain, or to have bacteria do the same. And actually they failed. Where Eduardo took one step back and said, what is the limiting step here? It's not nodules, it's not bacteria. It's actually, the reaction only works in low oxygen environments. And Eduardo figured out that bacteria can create those through what's called biofilms. They have a little envelope around themselves that they can make if you give them the right medium, and then they fix nitrogen for you. And about 15% of the bacteria in soils can actually fix nitrogen. They just don't have that right environment. And he found that plant compound, in this case, apigenin, is a compound that the plants extrude from the root, will attract nitrogen-fixing bacteria, induce them to create this envelope around themselves, and give them the environment they need to fix nitrogen. This is only a greenhouse assay, but it's, if you can see with Kataki on the left, that's the line that was gene edited. Again, he just knocked out a couple genes, and he has a couple different events And you could see the plants are shorter, but there's a lot more stems, there's a lot more grain in those plants. And he was able to also do this now in wheat, which is much more complex genetics. So these genes are actually ubiquitous in any plant we find. We can do this. We looked in tomato, we looked in pepper, we looked in lettuce. These genes are there in plants. So this is quite it sounds simple, but I mean, this is very doable in any crop, I believe, with the right people working on it. Next slide. We can use gene editing to look at problems with food safety. You think of aflatoxins, for example, or fusarium aflatoxins in wheat or in well, aflatoxins are really found not only in corn, but they're found in really most legume species, nut species, and so on. Salmonella, E. coli, especially in the leafy greens and things like that. Next slide. Gene editing is happening really around the world. There's about a dozen projects going on in Africa that I know the people quite well, in all sorts of different crops as well: bananas, cassavas, yams, sweet potatoes. These aren't crops the U.S. is really worried about, to be honest. So people are doing it right there, in their countries. And really, the U.S. is going to become a net importer, of products, unlike, say, in the GMO technologies. Next slide. I'm going to throw this up just to set up my next speaker, Chou. Next slide. I just want to show that. So green means have good biosafety protocols and regulation. I'm just going to say I'm putting US as yellow right now, and I'm going to leave it at that because it'll be explained in the next talks. Next slide. The one thing we need to have, and again, it goes back to my title slide, is we need good policy, we need good regulation, and the policy needs to address all the crops, really not just the big 5 crops that currently have GMO approved in them. They're your major big crops. I don't need to list them, you know them. Crops are complicated. They have, most of the time, their genes are actually duplicated, so the policies and regulations need to address that. Right now, they limit that. And gene editing is not only knockouts. We can insert things, as I mentioned, up to 25 genes right now. This is actually happening. I know Corteva is putting 8 genes together in one locus for disease resistances in corn using this type of technology. And, we know quite a bit about plants, and we just need to, I think, take this with common sense, and hopefully we can all use the tools. Next slide. And that's all I have.
Thanks for that, Alan. Very, very interesting stuff. We're going to jump into our next speaker. Maybe as I do that, I might remind folks, if you have questions, please put them in the Q&A at the bottom of the screen, and we'll get to those at the end, or our speakers might answer some of them on the fly. So if you have things that you want to know, don't hesitate to do that. Our next speaker is Dr. Rick Lawrence. He's been with Bayer Crop Science since 2008 and is currently head of genome editing, yield, disease, and quality research in plant biotechnology. He led the pioneering work at the company in gene editing and application of these tools to develop the first edited crop plants. He subsequently led teams that applied genetics and genomics to develop molecular markers for trait development, and also held responsibilities for developing next generation genotyping technologies and high throughput automation systems. So thanks for joining us, Rick. The floor is yours.
Thanks, appreciate the introduction. And also, I think, thanks, Alan, for the previous talk. I think it'll segue nicely into where I'm going to go, because you provided some great examples of where we can go in the future. I'm going to take a step back and talk about, from a product development standpoint, how we're going to start bringing some of these products to market, and in particular, how we'd like these to be looked at from the regulatory jurisdiction perspective, and then really conclude with a bit of where we're going in the future from a commercial standpoint. And with that, if you could move to the next slide. So, there we go. And with this build, maybe you could just click it a couple more times. Sorry about that. There you go. So, and Alan set this up But just to maybe reiterate the differences between gene editing and plant breeding and GMOs, all tools in the toolbox to develop innovative new products for farmers and consumers for the future. One of the key differences is GMOs. There you're accessing a gene pool outside of typically the plant, what a plant is sexually compatible with, to bring in a gene that's going to provide a new favorable characteristic. Whereas gene editing, you're going in and you're modifying the existing genes within the genome, optimizing those to create a favorable characteristic, or potentially removing an unfavorable characteristic. And then in contrast with plant breeding, which has been going on for thousands of years, there you're essentially shuffling genomes together, trying to bring together all of the many different traits that have randomly or spontaneously occurred in nature, and then creating progeny that have all of those characteristics that are going to be of benefit. So then if you go to the next slide, and at the end of the day, if you were to think about gene editing, what it looks most similar to from a final product perspective, it actually looks most similar to conventional breeding. So essentially what genome editing allows you to do is create variation within a plant that is going to end up looking a lot like conventional breeding. Now, a lot of times from genome editing perspective, the way you get there might or may not involve a transgenic intermediate. And so that's one of the key differences. And then if you go to the next slide. So one of the challenges that this creates is that how our various, and I wonder whether several of us will have pictures of the map. But in any case, one of the challenges that this creates is different geographies and jurisdictions around the world are looking at this differently, coming up with different ways to actually try to bring these products through their jurisdictions to market. So some have defined GMO exemptions, some have a modified GMO review process, some are case by case, et cetera, et cetera. And if you go to the next slide, what this ends up creating is essentially a long and laborious process to bring products to market. And what that does is it delays innovation. It's inhibitory towards your smaller players trying to innovate and bring products to consumers and to farmers. So that's kind of the current state that we're in. And then if you go to the next slide, yeah, one of the fundamental similarities for some of the regulatory policies that have started to look at genome editing that is aligned with where we believe it should be looked at is that genome editing should be looked at essentially as anything that can be should be exempt if the edits can be generated through conventional breeding. And if you go to the next slide, if you think about what that actually means, well, there's many different types of changes to genomes that can occur with conventional breeding or spontaneous, or that have occurred spontaneously in nature that are leveraged in the plant breeding process. And so things, I think some of the things that we typically think about are small single base pair changes or small insertions or deletions, but gene duplication, introgression from wild relatives, large or small inversions of genomic segments and translocations are all part of the breeding process. And some jurisdictions are looking at different aspects of these types of things that are leveraged in the plant breeding process as different. And so if you go to the next slide, a little bit more detail from this nice paper here that shows some of the different types of more specific changes that are utilized in the plant breeding process. And interestingly, things like insertion of bacterial genes and retrovirus types of genes inserted into genomes, or fragments of old agrobacterium tumefacients, insertions, transposal elements, et cetera, et cetera. So many genomes are dynamic, constantly changing naturally. And when you think about genome editing, you can create a lot of those similar changes. And even better with genome editing, you have the ability to really specifically change those. So then if you go to the next slide. So I'm a scientist. I lead a science group, a scientific program to utilize genome editing to generate our next generation of products. So why am I telling you all of that about genome editing and why we think it should be looked at as regulated exempt like conventional breeding? And the reason is because we believe that in its most fundamental, most innovative application of genome editing is in a way where you apply the technology in a way that you really bring together a breeding type of process with genome editing. And so one of the key innovations with genome editing and the CRISPR-Cas system that was invented in 2012, 2013 was the fact that one, it was fast and it was easy to design, which made it scalable. But also it allowed, that system allowed one to make many, many different edits at the same time. And genome editing had been invented in the 80s with zinc finger nucleases, and then it evolved into TALENs. But it was the CRISPR-Cas9 system that took off, that helped the technology take off because it's scalable and allows you to actually make many modifications. And when you think about what that allows you to do, it allows you to really go in and create a targeted variation in genomes that is most like what is currently used in breeding processes, except that you can go in and make very targeted and specific edits and variation at loci that you believe or you have hypotheses that will create new ideal performance characteristics. And then maybe if you just go to the next slide, I'll talk a little bit about why, from a product perspective, what we think that will allow us to unlock. And so there's a couple of key examples out there, and I'll start. If you think about the domestication of something like corn, which took thousands of years, and a few different key specific mutations were artificially selected on over the years. You know, the separation of the tassel from the ear and the ear placement around the height of where someone could hand harvest it, removal of tillering so that you have one single stem. All of that took thousands of years, but many different spontaneous mutations had to occur, right? And so there's a company called Covercress, and what they did is they took a weed, a pennycress weed that occurs naturally in North America, European fields, and within about 7 years and just a few edits, they turned that essentially into a new type of crop. So it's they've been able to remove the weediness. They've been able to modify the seed composition in a way that it is now suitable for meal, and the oil profile is suitable for a sustainable jet fuel type of value stream. Not to mention it's a cover crop, so it can be utilized to provide a carbon benefit to the soil and to the farmer. So really cool story, just to illustrate what we think genome editing can unlock when you're able to actually make multiple edits to the genome. What it really allows you to do is create new crops, rapidly adapt crops or domesticate crops. And just one other example like that, that's a little bit more like the consumer example is Pairwise recently. I think many of you know the story of brassica species and the consumption of brassica species. So the thousands of years that led to things like of domestication that led to things like broccoli and cauliflower and kale and Brussels sprouts, et cetera, et cetera. Well, what Pairwise did is they essentially created a new type of brassica crop as well in just I think 3, 4, 5 years. And what they did is they took a mustard green that has a really pungent flavor, but is really nutritious, so not really something that people want to eat. And they modified it. They removed, I think, 17-ish different genes, don't quote me on that, and removed the pungency, horseradishy type of flavor. And now they have a leafy green that has a similar nutritional profile to like a kale. However, it has the crunchiness of a romaine. So again, really cool story about the ability to create something new and novel for consumers really rapidly with this ability to modify many different genes in a genome. And I think that's just scratching the surface. A bit of what you'll see in the future from a product perspective is going to be in that realm. So if you think about what nature does not do well, nature does not it's really slow to create spontaneous mutations that are useful, but two, many mutations or combinations of mutations that are useful are that's even slower and poorly sampled in the environment. What editing allows you to do is actually create that same type of variation, or create a more targeted type of variation towards performance characteristics really rapidly, that we think is going to allow us to actually accelerate our ability to, one, design new crops, and two, accelerate the performance characteristics of the staple row crops around the world. And so with that, I can conclude and turn it over to the next speaker. Thank you.
Thanks for that, Rick. Very interesting. We're going to go on to our third speaker, Dr. Fan-Lee Chou. She's a Senior Vice President for Scientific Affairs and Policy at the American Seed Trade Association, where she leads ASTA initiatives on plant breeding innovation, intellectual property rights, and domestic and international regulatory policies. Prior to ASTA, Fan-Lee served at the USDA for over a decade, including as Agricultural Biotechnology Advisor, the Office of the Secretary, and its positions with the Foreign Ag Service and the Animal and Plant Health Inspection Service. We look forward to hearing what you have to say, Fan-Lee.
Thanks, Jason, and thank you for the opportunity to speak with you all. So next slide. I think we can go on one more slide, please. Just a little bit about ASTA. We are a association that was founded in 1883, so around for quite some time. We represent a broad range of the seed industry. We would like to say alfalfa to zucchini, and our companies support production across the board. We support biotech crops, conventional crops, as well as organic production. So our over 700 members are involved in a range of plant breeding, plant innovation, plant seed production, distribution, as well as all the adjacent services that's around the seed breeding and production process. Next, please. So Alan and Rick talked a lot about plant breeding and genetic diversity. So this is kind of my pictogram of what that looks like. So when we look at a carrot in the supermarket or a pepper in the supermarket, there's actually quite a lot of genetic diversity within the plant species itself. And for many of our members, the idea is how to use genome editing and other plant breeding tools to really leverage that innate genetic variability, and also to use these tools to increase or to induce genetic variability. And so these genetic variability are things that you can see and things you can what we call a phenotype. And there is a genetic basis for all the varieties of the phenotype we see. Next slide, please. So leveraging this genetic variability around a plant variety, plant breeders use a bunch of tools. And this is not my illustration because I didn't design this slide, but a slide of how plant breeding and the tools of plant breeding has evolved over time. And as you can see, the road gets wider as a function of time. And for me, that means that plant breeders and the plant breeding community are accumulating different types of tools. And we are not replacing certain tools. We're using a lot of these tools in conjunction with each other. And as we go along this row of plant breeding, the scientific underlines of how a phenotype is conferred by the genetic basis is better and better understood for a wide variety of plant varieties. So these tools are now becoming more and more precise and more predictive. So the whole idea is for plant breeders is to use these tools and use this genetic variability that can be naturally occurring, spontaneous occurring, or induced by some of these tools to really make plant breeding a lot more efficient and predictable, right? So in the beginning, when we were just looking at different varieties and picking and choosing based on observable phenotypes, it is kind of it's like a numbers game. The more you pick, the more kind of combinations you have, but you don't really have, as a plant breeder, control over that combination. So, but with new technologies like genome editing, like marker-assisted breeding, you can actually have a little bit more control, increasing control about how you make those combinations, and the outcome is more predictable. So you don't need as many numbers, so like thousands of plants or hundreds of thousands of plants, to actually find the combination or put together the combinations of desirable characteristics that you want. So next slide, please. So as we're talking about kind of, and I apologize to Allison because we have 3 plant persons and it's the 1 animal person. So I'll try to make allusions to some of the plant, the animal applications as well. So as we're talking about whether genome editing can bring new players into this space, as Alan asked, is there room to play? There are, I think, three-legged stool to decide whether it's going to be a stable position or not. And one of them is research and development. Is the science there? Can we do the science? The second one is there going to be market acceptance? And what I'm going to talk about is more the regulatory space. Next, please. So this is very plant focused, but I think it applies to animals as well. So as I said in the previous couple slides, we have this continuum of plant breeding tools that's used to develop products, new plant varieties. So for all of these plant varieties, regardless of whether the tools you use to develop them, there is a compilation of regulations that governs when something is placed on the marketplace. So for plants, in a lot of countries, you have to register the variety with certain competent authorities. There are seed laws in the US. We have the federal seed law that dictates how you can market and label those seeds. We have phytosanitary requirements that protects our agriculture, our natural environment from pests and diseases. We have general environmental safety laws. We have food safety laws and feed safety laws. So these are applied kind of technology neutral. And I would say this would be the same in the animal breeding space. You have these general laws and regulations that applies regardless of what kind of breeding methods are used. But we have one very specific regulation that is technology specific. So when gene editing, so this is GMOs, transgenic technology, came aboard back in the 1980s, we in the United States put together, the government developed the US coordinated framework to regulate products that were developed using biotechnology. And then many countries established laws in their own country based on the Cartagena Protocol on Biosafety, and that the Cartagena Protocol on Biosafety is an international environmental treaty that looks at the environmental release of biotech products. So these two kind of international and domestic regulatory frameworks are very technology specific. So when gene editing came online, people were asking, are products gene editing GMOs? Are they going to be subjected to GMO regulations? And the answer to that question is actually really important because it will determine who will have access to this technology to bring products to the marketplace. And as Alan and as Rick talked about, most applications of gene editing is to create or to compile genetic variability that occurs within the plant genome. And this is something that from a conventional breeding perspective, we've been doing for generations and centuries. So from ASSET's perspective, next slide, please. And from the perspective of the international seed community, so this is under the International Seed Federation, our underlying principle is that if a plant variety is developed through the latest technologies, such as genome editing, it should not be regulated differently if it is similar or indistinguishable from varieties that could have been developed through conventional breeding or be found in nature. So the reasons so in a nutshell, this is kind of a very wordy way of saying that if the product is similar to what could occur through conventional breeding techniques, then it should be treated the same under the regulation. And I would say this applies to animals as well as to plants, this underlying principle. And the reasons for that is because the safety profile of that plant is not based on the genetics, it's based on the phenotype, on the characteristics it expresses, right? So if the characteristics are similar, then the safety profile is the same. Therefore, there's no additional need for this extra kind of technology-specific regulation. So many countries around the world are thinking about this question, about how they will view genome-edited products, plant products, in kind of the context of their GMO regulation. Next slide, please. So this is my map. This is the map that the International Seed Federation maintains as we're monitoring the regulatory policy around the world. So the green in this map indicate that these are the countries that are making a regulatory policy decision that certain gene-edited products are not GMOs, right? On the other kind of extreme, and I apologize to those folks that are blue-green colorblind. I don't have another. This is the ISF colors. So the red color, there's only 2 of those, are countries where all gene-edited products are GMOs. So, and then in the middle you have the light green, and these are places where there are proposed policies in place that differentiate gene-edited products from GMO products. And the purple are countries where it is all gene-edited products are GMOs, but certain gene-edited products have a lighter regulatory approach than the full GMO regulatory assessment. So I just want to note, in Europe, it's kind of, yellowish green, and that's because the proposal that the Europeans put forward is a combination of both. It's a combination of differentiating certain genome-edited products from GMO, and then also including other types of gene-edited products and their GMO. It's quite a complicated proposal that they have put forward. But I think the take home from this is that more and more countries are recognizing that there is a difference between gene-edited products and GMO products, and they're reflecting that in their regulatory policy. For many of these countries, say in the South Americas, Brazil and Argentina, and even in Japan, this regulatory policy applies to plants and to animals and to microbes. So their regulatory policy is for gene-edited organisms, right? In other places, like in the US, in Canada, and in Europe, the proposal or the policy right now only applies to plants. So they're considering a policy for animals as well. So that is to be determined. So while in general, the overall picture is encouraging that countries are making that differentiation between gene editing and GMOs, the implementation is still herky-jerky a bit, right? So where the rubber meets the road, it is not completely smooth sailing, and we can talk about in the Q&A. Next slide, please. So just some general observation from an international perspective. There is growing alignment around international regulatory agencies that not all gene-edited plants, in some places animals, should be treated as GMO. And their determining criteria is in the final product, whether there is foreign DNA or not. If there's no foreign DNA, then that product is not GMO and it is conventional. And so all the other regulatory oversight that I talked about that is technology neutral still applies. So many countries takes a case-by-case approach, meaning you come to the agency and you go through kind of a verification and review process. And many countries allow for early consultative process. So even in the product development or product conceptualization stage, this is very, very important for small companies where the GMO or non-GMO determination will make or break whether they go forward with a product or not, because they most of the time cannot afford the full GMO regulatory cost that's associated with the GMO designation. And as the first countries that went out of the gate with these policies, Argentina and Brazil, they are making concerted efforts in the South America region and in the Central America region to harmonize their approach. And it's kind of easier to do in that region because they have very similar legislations that governs these regulations of these products. Next slide, please. So I just want to spend a little time to talk about the US because this is where CRISPR and Cas9 invented, were discovered, and it's really important for us to be able to use this technology in the US as well. So in the US, we did not pass a GMO regulation or an overarching GMO or biotechnology law. What we did was put together a framework, the US coordinated framework. And that framework used existing laws that USDA, FDA, and EPA doesn't really apply to animals, and looked at the intent of those laws and apply those to gene-edited products. So the products of biotechnology were regulated based on the intended use. So for example, if you're going to create a plant used in food, then FDA has jurisdiction because they're the food and feed regulatory authority. If you're going to create a plant that has a plant protectant, like a pesticidal intent, then EPA has regulatory authority. But if you're going to plant that plant anywhere in the US, import it into the US, or move it across state lines, then USDA have regulatory authority. It's slightly different for animals, and Allison will talk about that. Next slide, please. So just to kind of just to go over a little bit how these three agencies, they were tasked by multiple administrations to clarify their regulatory policy in light of gene editing applications in plant breeding and also in animal breeding. So USDA was the first to come out with their revision back in 2020, and they created these exemptions, what I would call kind of conventional-like exemptions. So that's hard for me to point, but in the big green box is these modifications to a plant that could have occurred through conventional breeding. But conventional breeding is really has a lot of different genetic variability, right? This is what Rick talked about. USDA focused on three. So this is their first exemption out of the gate, that if you make a single base pair, a single targeted base pair substitution, that's number two. Number one, if it's a repair without any template, you just create a cut and cell repairs itself, and these usually cause deletions and insertions that kind of changes the expression of that gene. Or if you introduce a gene known to occur in the plant's gene pool, those three alone, applied singly, would be exempt from USDA regulation. You cannot use them in combination or so those are kind of a very narrow subset of what could occur through conventional breeding. But what USDA recognized that in their rulemaking, so they added this component that in the future, they would add additional categories for this exemption to kind of widen that exemption or bringing in more conventional-like exemptions into that space. But what USDA also did that was really kind of innovative from a regulatory perspective is moving away from GMO regulations, this event-by-event type of regulations. What they did was that they looked at the products that they have reviewed. They looked at the plant trait mode of action. So how does the plant got its trait? And for anything that they've reviewed before where they found this to be not a risk to their protective goals, then those things, that plant trait mode of action is then exempted from regulation. So this applies to gene-edited plants. It applies to traditional transgenic plants. So for example, if you have a corn that has, say, cry1A protein and it gives this insect resistant, USDA reviewed it, deemed it not regulated because of lack of risk. And then anyone who creates a corn cry1A that has the same insect resistant is no longer regulated. That product is no longer regulated by USDA. So this list of plant trait mode of action continues to grow as USDA is reviewing more and more crops. So USDA have a regulatory status review process for plants that doesn't fit into these exemption categories. And as USDA make determinations of not regulated from a regulatory status review process, it gets put into this plant trait mode of action. So that list is continuing to grow. So you can go to the USDA website and look at products that have gone through the regulatory status review process, the plant trait mode of action exemption table. So any of those combinations of plant trait mode of action that can be exempted. And they also have this voluntary consultative process where if you believe that the plant you have created is exempted based on any of these categories, you can go ask USDA for a confirmation. So there's these confirmation letters that you can look at for products are exempted from USDA regulation. Next slide, please. So EPA was the next agency of the tri-agencies under the coordinated framework that put out a revision. This was just recently in July 2023. So EPA does not regulate plants, right? They regulate the plant incorporated protectants. So the substance that the plant produces that gives it that pesticidal protectant intent and the genetic material that is inserted to create this. So this rule was written kind of with transgenics in mind. So the revision from USDA, from EPA really wanted to reflect the new applications of genome editing where you're working within the plant's genome. So when EPA put out this rule, their first rule back in the days of transgenic plants, they recognized that plants create their own plant protection, right? From a conventional breeding perspective, we use plant breeding to create plants that are resistant to plant pests. So initially, EPA had created this category of PIPs, plant incorporated protectant, developed through conventional breeding. So when they revised the rule, what EPA wanted to do is to reflect that you can use genome editing to actually create PIPs that are like conventional bred PIPs. So they created two categories of these PIPs to be exempted. One is loss of function. So using genome editing tools or other plant breeding tools to create this loss of function where you are removing a substance but the plant then gains a protective characteristic. The second category that they propose is these PIPs that is developed through genetic engineering like gene editing, but working within the sexually compatible gene pool. So kind of equivalent to the conventional PIP exemption, but they created certain, I would say, criteria around this so that it reflects what is already done through conventional breeding. But for both of these, there is a mandatory notification to EPA, there's a mandatory record keeping, and then for the PIPs created through genetic engineering from sexually compatible plants, there's a verification process that EPA has to go through before you can put your product in the marketplace. So unlike USDA, where you can only do one of the kind of conventional exemption at a time, EPA doesn't have a limitation. You can create multiple PIPs within one plant, and they will review them to see whether they're exempted or not exempted from their regulatory process. If your plant does not fit into any of these categories, then it will have to go through full registration process. Next slide. So just looking at the two agencies now that have put out their revision with the view of gene editing applications in plant, there is pluses and minuses to both, but there is inconsistency across the board. So there are two circular diagrams, if you will, of what plants could be exempted. There is some overlap, but there is majority of lack of overlap. So for USDA, you have a voluntary confirmation process. You can only do single modifications. So only single modifications are exempted. And the exemption does not apply to multiplexing of traits. But in the future, USDA would add additional exemptions. So theoretically, they can address these shortcomings. On the other hand, EPA has a mandatory notification and confirmation process. It limits the kind of the range of genetic variability you can introduce to things that are identical to what has already been done through plant breeding, right? So it doesn't allow for the creation of new genetic variability, only a copying of existing genetic variability. On one hand, that's quite limiting, but on the other hand, it allows a plant breeder to multiplex multiple edits, multiple characteristics into one plant. But there is no easy way for EPA to add additional exemptions or change the criteria for exemptions. They will have to go through rulemaking. So the last agency I want to talk about is FDA. And FDA, we're still waiting for them to clarify their policy. So FDA look at food, right? So for gene editing, we're actually working with endogenous substance within food. We're not adding any substance. So in that space, the requirement for plant breeders is to ensure that the endogenous substance are within safe levels. There's actually no mandatory requirements to go to FDA for this, but we are waiting for FDA to kind of come out with their guidance on how they may view food and feed derived from gene-edited plants. Next slide, please. One more, please. So the take-home message from the regulatory landscape is that internationally, the trend is to differentiate genome-edited plants from GMO regulations. The pathway to market is getting clearer, but there are still quite a number of challenges. So there's regulatory schemes that are quite untenable in certain markets like China, and Europe remains to be seen. In China, they view all gene-edited products as GMOs, but certain gene-edited products can have a more streamlined regulatory process. There are differences in implementation, even in the US, but across the world, there's differences in the information they require to make decisions and the timeline they take to make these decisions. What's interesting is that in this space, the regulatory kind of leadership is not the same as in traditional transgenics. The folks that are taking the lead are South Americans like Argentina, Brazil. In Asia, it's Japan. In North America, it's Canada. So it remains to be seen like how this, I think internationally that the regulatory environment is becoming more and more enabling for innovation. And we will have to see what kind of products will come out from this space. That's it for me. Thank you.
Thanks for that, Fan Lee. As was indicated, our 3 prior presentations primarily focused on plant agriculture, and our last but certainly not least presentation will, I think, shift gears a little bit and focus on gene editing applications in animal agriculture. So that last presentation will be by Dr. Allison Van Eenennaam. She's a professor of cooperative extension in the field of animal genomics and biotechnology in the Department of Animal Science at the University of California, Davis. Her publicly funded research and outreach programs focuses on the use of animal genomics and biotechnology in livestock production systems. Her current research projects include the development of genomic applications to select for cattle that are less susceptible to disease and development of genome editing approaches for livestock. She serves as USDA NRSP 8 Cattle Genome Coordinator and was a member of the National Academies of Science, Engineering, and Medicine Study Committee for Science Breakthroughs to Advance Food and Agricultural Research by 2030. So I'll turn the floor over to Allison, but before I'll just remind you to please put any questions you have in the Q&A at the bottom of your screen, and we'll get to those as soon as Allison finishes. Welcome, Allison.
Okay, thanks, Jason. Can you just give me a thumbs up that you can hear me okay?
You're all good on my end.
Okay, very good. All right. Well, thank you. And I feel the weight of the animal kingdom on my shoulders because we've had 3 discussions around plant breeding, but of course, the other kingdom of importance to agriculture is the animal kingdom. And we have similar interest in using genome editing. And I will just go over kind of what it's been used for. But before I do that, I just want to say that although we've had traditional GMOs or genetically engineered plants that have been widely adopted by millions of farmers throughout the world, the use of that technology in animal production has been very limited. In fact, there's only 2 approved food animal uses for the technology, and of course, that famous aqua advantage fast-growing salmon. But really, the costs and regulatory approaches that have been used for the technology made it very unattractive to utilize this technology in animal breeding programs. And a lot of our focus today has been on regulation. You might wonder, my God, why have all these scientists talking about regulation? And the reason is because if the regulatory approach is so expensive and difficult to maneuver, it effectively precludes innovation and the use of that technology especially by smaller companies and academics. And so it is a really important, I guess, enabling technology that we can use these tools and that the regulatory doesn't effectively preclude the use of the technology in animal breeding. And so If I can move to the next slide, please. I'm going to go over a review that a postdoc of mine did, Alba Ledesma, of the published uses of genome editing in the scientific literature. And I'll just kind of start from the bottom here to the most, the targeted trait that is of the most interest, and probably no big surprise there that that's yield. And this is broken down by different categories of animals. And so the ruminants, for those plant people that maybe don't know who's who, that's the sheep and the goats and cattle that have a rumen, or stomach chamber that can digest cellulose. Then we have the monogastric mammals, and there we're mostly talking about pigs, also rabbits. Then aquatic animals, and that's a very big category, very large fish consumption throughout the world. Then we've got avian, mostly chickens, and then insects at the bottom there. Then the next thing you'll see is reproduction. And so you might go, well, that's an unusual second most focused trait. And the reason for this is really altering breeding program design to some extent. And so there's a lot of work by researchers in basically all of those different kingdoms, or not kingdoms, but types of animals, to develop animals, for example, that are infertile. So for example, in salmon aquaculture, you might want to have fish that are commercial, but are not able to pass along their genetics to wild populations. And there's also some other reasons why you might want to pursue reproduction in these different groups, and things like single gender or sex offspring. So you only want females in laying chickens, for example. And so is there a way that you can have chickens that only produce one sex of egg, i.e. females, or a way to basically identify the male eggs so that they don't go through hatching because they are not really the target animal in the case of laying chickens, for example. Biotic stress means disease resistance, very big focus, and I'll go over a couple of examples of those as I go through the different examples in this talk. Hypoallergenic would be knocking out the proteins that make people allergic to, for example, milk or eggs. And so, of course, you can use genome editing to go in and very specifically target and knock out those particular proteins. Maybe the alpha-gal that gives certain people allergies to red meat. Those are the types of traits there. There are, of course, now groups that are wanting to stack traits, which is using the nomenclature of the plant people, i.e. I would like a disease-resistant, hypoallergenic animal with good yield that is maybe sterile, so it can't reproduce with wild animals. So basically combining those traits. Color is really a big focus on some of the ornamental fish, as you could imagine, where that's a very important characteristic. Others are kind of don't easily fit their quality, is trying to improve the product quality. And then finally, at the top there is abiotic stress, which is things like temperature tolerance or ability to withstand high temperatures. And in that other category includes some things for animal welfare, such as the fairly well publicized example of dairy cattle that would have an allele that prevents them from growing horns, such that they don't need to be physically dehorned, but can be genetically introduce an allele that exists already in some breeds of beef cattle like Angus, and introduce that form of the gene into dairy cattle so that those animals don't grow horns themselves. So that's kind of the breakdown of where it's getting used. And the next slide, if you will, is a rather interesting graph of where those publications were coming from. And you can see there's a very clear leader in terms of the development of genome-edited animals, and that's China in terms of publications. And they really are working on the whole gamut of the different types of animals. So the ruminants, monogastrics, aquatic, avian, and insects. That's followed by the United States and Japan. I'll get into a little bit. Maybe is unexpected to see them there, but they're actually doing quite a lot of work and have had actually some work in fish that I'll discuss in the next slide. Then there's a smattering in other countries throughout the world, but very clearly this is a real emphasis and focus of Chinese scientists. So if I can go to the next slide, please. This is a smorgasbord of all of the different examples of different types of animals, and then broken down by species, and then where it's getting used in those species. And so the winner here is pigs. So they are the most targeted animal, at least with regards to publications that are out there. And that's a little bit because pigs, you can edit their embryos and get a litter-bearing animal. So you get lots of piglets, which is much easier to work with than, for example, a cow, where you get maybe one calf a year. And so you can do work a little bit more rapidly with pigs. And also they're a very important food species globally. And then you can see the cattle and goats and rabbits. And then, of course, aquatic animals includes a whole range of different species. And so they are really nice because you can introduce the editing reagents in the eggs after they've been released by the female, and you don't need to then replant that egg back into a synchronized surrogate dam or recipient, which you have to do in mammals, where basically you edit the embryos typically, before 7 days of after fertilization, and put it back into the pig or the cow or the sheep. So it's a little bit easier to do it in aquatic animals. And so you'll see there's two there that are highlighted in green. And those two are tiger pufferfish and red sea bream. And if we can go to the next slide, the reason that they are targeted is highlighted there in green is because these are approved genome edited fish that are actually in the market in Japan. So they're being sold to consumers. So the regulatory agencies in Japan considered these two fish, both of which are knockouts. In the case of the bream, it was a knockout of a gene called myostatin, which myo is muscle and statin is stop. If you knock out the myostatin gene, you get a muscle go, if you will. So you get a increased muscle yield. And you can see that in the image below or to the left of the Japanese flag. And similarly, the pufferfish has had the leptin receptor knocked out, so it has a bigger appetite, if you will. And so that's the first products of animal biotech or animal genome editing that has made it to market. And then on the right, I have an example that was produced by the Aqua Bounty company, which is the company that produced the Aqua Advantage salmon. They're an American company, where they knocked out, again, myostatin, in this case, using CRISPR-Cas9, and produced tilapia that have an increased muscle mass. And they have been determined by both Brazil and Argentina to be not a GMO. They're not containing foreign DNA. And so they have determined that these animals would not be subject to the GMO regulations in those countries. And both of those countries have an approach where you can go with a product design or a concept and ask the regulatory agencies, if we produce this, would it be regulated as a GMO? And then they'll give you their determination. And that gives the developer some certainty as to moving forward, whether they're dealing with a situation where they have to go through a full GMO approval or whether they're going to be treated the same as conventional breeding. And so this particular fish, the myostatin knockout, was judged to be non-GMO. I will say that they're not commercializing that at the moment because basically it wasn't an elite line of tilapia that was edited. And so if they did want to commercialize it, they'd go back and actually edit, very elite lines as you would with any breeding program. And that's an important point, is this has to fit on top of a breeding program. And I appreciate the previous speakers kind of emphasizing that it's not a standalone technology. It's going to be something that would be in addition to an existing breeding program. So working with the very elite germplasm at the top of the breeding pyramid. So if I can go to the next slide, there have been sorry, I should kind of give an overview here of the different regulatory approaches that have been taken for genome editing. And this is from a paper by Eric Halleman. I'm in 2022, and I guess this is my map. Everyone's had their map. And what you see here is similar to plants. So at the moment, EU and New Zealand have kind of a GMO-only policy. And then other countries such as Brazil and Argentina have regulatory policy with exclusion. So basically, if you don't have any novel DNA, you're not treated differently to conventional breeding. And then you'll see the kind of hashtags in the case of the United States and Australia, and I'll get into what that is. But basically, the United States is regulating any edited animal as a new animal drug, with the possibility of enforcement discretion, which is a kind of a nerdy term that I'll explain in a minute. And Australia is regulating certain types of edits as GMOs if they've used basically a template for the repair, and I'll kind of discuss that as we move along. So Brazil and Argentina, to kind of emphasize Van Lee's point, are leading the way in terms of developing policies that look at what has been done and determine whether or not it's going to be regulated as a GMO. And so if I can go to the next slide, there have been cattle that have been using simple modifications. So basically these are knockout cattle in the case of these cattle, where there's animals that have had their myostatin gene knocked out. So Nelore cattle there on the right with increased muscle yield, and then cattle that have their prolactin receptor gene knocked out using genome editing. Both have been considered to be non-GMO by Brazil, and they're both produced by the US company Excelogen. And if we go to the next slide, we can see that Argentina has made similar determinations around slick editing. So that's the prolactin receptor that gives the animals a slick hair coat that helps them tolerate very high temperatures. And they also have a product concept. So this has not yet been produced, but to produce an animal that is both slick, Holstein, in addition to having the allele that makes them not grow horns. So this would be an animal that would have both the heat again. Argentina has said, because there's no foreign DNA sequence and as such no new combination of genetic material, they would be considered non-GMO. And so that again is a product concept that they're running through regulatory and getting a determination on before they proceed with developing the actual animal. And then next slide, last year, it was determined by the had these knockout animals where they had basically knocked out the prolactin receptor to give them the slick characteristic. So this is a naturally occurring mutation that occurs in certain breeds of cattle, Senapol, for example. And they took that to the FDA and said, look, here's these two animals. We've done this modification. We've sequenced them. Here's the knockout. The slick's been knocked out. Can we move to market with them, basically? And what happened was FDA gave them an enforcement discretion to these two particular animals. And what that means is that the FDA evaluated the animals, determined them to be low risk, and as such, they are exercising their right to not enforce the rules whereby you need a new animal drug approval to bring an edited animal to market. And this is called enforcement discretion. So it's exercising that right. In this case, it's for those two particular animals, and they are allowed to go to commercialization, their milk and meat, and also their reproductive products such as semen and eggs. And so there has been that determination. It's not technically an approval, it's an enforcement discretion for those two particular animals. And if I made the same animals, I would have to go through my own evaluation. It doesn't give a blanket exemption for all slick animals created from genome editing to go to market. And then I'll go to the next slide and discuss a little bit the product that is the first one to go through the full regulatory process at the FDA. And this is a knockout, so no novel DNA in this case, of a gene that results in pigs that are resistant to a disease called porcine reproductive and respiratory syndrome virus. This was first developed by the group at Missouri to knock out this particular gene to give animals that were resistant to PRRS. It was subsequently worked on by groups at the Roslin Institute and Chinese groups. And basically, this is a widespread disease that affects all of the orange areas on this map that is very deadly to pigs and costly to pig farmers. And ideally, you'd have pigs that didn't get sick. And by knocking out one particular little section of the gene, you remove the site where the virus can attack the pig, and you get a pig that's just no longer susceptible to that particular virus. So a company called Genus, which is a publicly traded UK-based company, is taking this through the regulatory process. And next slide, please. And I think this is an interesting difference between the slide that was shown in plant breeding, and this is how we kind of do animal breeding, and it's almost an inverted pyramid relative to plant breeding. So basically, we typically have a four-way cross for pigs. So there's sire lines and dam lines that are great-grandparents that basically are multiplied up through the breeding pyramid to produce the commercially produced animals. And so you can see here one great-grandparent is basically producing thousands of animals down at the harvest level. And so if we want to introduce a mutation into these grandparents, we're going to have to target all of the different grandparental lines and get it into a homozygous condition, i.e. both alleles of that gene will have to be knocked out using editing in each of those grandparent lines. And to maintain genetic diversity, it can't just be one animal of each grandparent line. It's going to need to be multiple animals of each grandparent line. And so that is quite a big ask. And I think it's important to realize that even if we have the technology, it still has to be integrated into our breeding programs. And so next slide, the way this is being approached is that they are introducing the edits into developing embryos to knock out the gene. They're then producing, putting those eggs back into surrogate or synchronized pigs. That'll give birth to a range of different piglets, and then the company's going to go through and select the animals that have one of two desired edited alleles, so heterozygote or homozygous animals. They'll go through then, if you go to the next slide, and categorize those animals, make sure that they are in fact disease resistant, but just as importantly, that they are still genetically elite, they still produce pigs, they're fertile, they're everything else that you would want in a commercial pig breeding program. They're going to phenotype that and make sure that all of that is as expected, so that there's no effects that are other than being disease resistant. And so they estimate they'll need 10 to 20 high genetic merit cross bores across the two maternal and two paternal lines to maintain a small nucleus population that's knocked out for both of those alleles. And that's what they're seeking regulatory approval from. And then once they get that, then those founders will be used then to multiply up those pigs across the breeding pyramid and produce the kind of the thousands that are down at the commercial level. So that's all detailed by Genus in a paper that I've cited there. And so you can see that that's quite a big undertaking, quite an expensive undertaking. And given the fact that these are unapproved animal drugs, all of these experimental animals aren't allowed to enter the food supply. And so having that type of enterprise running and having no saleable product really makes it very, very costly to obtain approval for that type of a product. So we have kind of a funny situation, and I'll go to the next slide, where if you have a knockout in a food animal introduced by genome editing, whether or not you're subject to additional regulations beyond the kind of traditional animal breeding regulations and the need to have a safe product really depends a little bit on which country you're in, and it's almost a coin toss at this stage. So you basically wouldn't be treated differently in Argentina, Australia, Brazil. Canada has a kind of a novel product risk approach, so it will depend a little bit whether this is a novel product that you're producing. Whereas in the EU at the moment, New Zealand, the US, you would have to go through full GMO approval in the case of Europe and New Zealand. And the US, you could potentially get enforcement discretion if you're a developer, but it's not going to be similar to what happens in Argentina and Australia. And then there's only one change. This is knockout, so you're inactivating a gene. If you're using a template to introduce an allele like the polled allele from one breed of like Angus into Holsteins or dairy cattle. Next slide, please. The only difference on this is now Australia is treating that at the moment as a GMO because you've used a DNA repair template. And that's really because of the way their regulatory language reads. And so it's a little bit of a difficult situation with such discrepancies and non-conformity between the different countries. And of course, animal products have traded throughout the world. And so if there's not harmony in the regulations, we have a potential trade disruption when it's not treated differently in Argentina, but then maybe they want to, export to EU or whatever. So if I can just conclude, next slide, please. I guess to me, I see editing as kind of the cherry on the top of the breeding sundae, if you will. So we are not going to remove our breed associations. We're still going to have breeding goals as a most important fundamentally outlining what we're trying to achieve. We'll still do performance recording and progeny testing and artificial insemination and embryo transfer and genomic selection, all that good stuff. But we have this new tool to bring in a useful genetic variant, like not growing horns, like slick, like disease resistance. And we can do that without having to cross an animal from a different breed and bring in all the unwanted genetics from that, something known as linkage drag. So we can do it in a very targeted way and not introduce genetics we don't want in the program. So in summary, next slide. Genome editing offers an approach to introduce useful genetic variation and alleles without linkage drag, typically associated with crossbreeding. Next, please. Scaling of useful edits to commercial livestock breeding programs will be technically complicated and expensive. I hope I showed you that in the pig example there. In many countries, simple edits like knockouts, moving an allele from one breed to another, where we haven't introduced foreign DNA, are going to be regulated as GMO. And I think this seminar series has made it very clear that the fate of genome editing is really going to depend on developing a regulatory framework that's risk-based and hopefully somewhat concordant amongst the different countries of the world and especially amongst trade partners. So with that, next slide, I would like to thank you for inviting me and having the animal kingdom represented on this seminar series.
Thanks for that, Allison. So as I've already encouraged you to do so far, you're welcome to put questions into the Q&A box. And I forgot to say this earlier, but it'd be nice if you indicated where you're from, maybe where you worked in your question. I might jump in with a moderator's privilege, perhaps, and ask a question to get the ball rolling. And that's, many of you mentioned on the panel different benefits that gene editing could convey. I think Alice, in your presentation, focused a lot on how much emphasis was on yield. I guess my question is really about if what other examples you all have really on consumer facing traits that really might change the game in terms of how consumers think about some of these technologies. I just wonder if you could say a little bit more about both the technology itself and maybe also how y'all might think about some of that consumer interfacing.
Well, I can start that. So yield is one of the traits, but I disagree that was only one. So there was hypoallergenic products, there was animal welfare products, like not growing horns. There is product quality traits that are being done to alter the quality of the milk and meat. So I think that there are consumer-facing traits and ones that align with values such as single-sex chickens for the layer industry. I mean, I don't know what who wouldn't want that really? And disease resistance, again, I realize that's not necessarily consumer facing, but not having sick animals is a real societal benefit in terms of wastage and use of antibiotics, and good for the environment, good for the farmer, good for the animal. It's a win-win. You don't get that very often.
And Jayson, if I can add, just from Asta's perspective, we have such a diversity of companies are involved in this space. And I think what makes genome editing exciting and the change in the regulatory policy exciting is that it creates more space for people to actually produce plant products that are more has direct consumer attributes. I would say that, all the things that folks are working on, like Allison says, has consumer benefits. It's just not immediately evident, right? So yield gains has consumer benefits. More efficient use of water inputs have consumer benefits, just broader societal benefits. But if you look at, and I think Europe, there's a university in Europe that tracks the kind of R&D development in gene editing in plants, you can see consumer benefits like non-browning avocado. That's my dream plant. You can see more nutritious mustard greens, like pairwise, their entire product portfolio is focused on nutrition and ease of use, like making things snackable is their tagline. So for, parents like me that's packing lunch, things that doesn't brew easy, that still tastes good. So there's a lot of things in the pipeline. So I think for us, a lot of people are like, oh, the regulatory policy is kind of clearing up. You should be seeing gangbusters of stuff coming out. Well, it's not that easy to bring a product to market, right? Regulatory is one thing, science is the other. So I think we're going to see things coming online. Like Allison was saying, in Japan, there's definitely a huge push, and their regulatory policy is super clear for plants and animals. So now it's just waiting to see whether you're going to put out a product that excites consumers. And not every product's going to be a game changer, but I think there's going to be some.
Yeah, thanks, family. One of the benefits was mentioned was in the first presentation that you gave, Alan, about nitrogen fixation. And there was a question that came up in the chat by Daniel Kaiser, and he asked about, who owns that technology? How's that commercialization will happen? And so I just wondered if you could weigh in on that. I know you answered in the chat, but for the broader group, and just maybe speak a little bit more broadly about IP and commercialization, and as somebody that works in the public sector, how you think about these issues and engage with the private sector to get these in the hands of producers.
Well, this is one of the technologies that, to me could be a game changer. And I'll go back a bit to the last question that I think everybody tried to emphasize. A consumer trait is the environment these days, right? People will say, oh, I'm saving water, or I'm saving nitrogen, I'm saving, resources somehow. And so I think that's changed since, say, the original GMOs, where people always thought, well, is it going to be a more flavorful something? Or something like that. But there's a whole, I think, segment of the population now where, this makes a difference. And nitrogen is a big issue worldwide. In nitrogen fixation is, most people think of nitrogen use efficiency in plants as opposed to getting nitrogen from the air, like legumes. The technology, I think, is very exciting to me. It's one of the most exciting technologies, I think, for climate change. And the fact that it's really a single gene edit. There just happen to be 2 copies in rice. And rice is one of the simplest genetics. It's a diploid. But Dr. Blumall is able to do this. It actually works even better in wheat, and which has 6 copies of everything. And actually, this one has 9 in this case. And he's knocked them all out. And it works. And as far as IP, UC Davis plays the game like everybody else. We're very collaborative. With companies. This technology was actually developed with a company, and I'll keep the company name out. But the idea is to license it. Davis has, for quite a while now, for the last 20 years, done what we call, it's through a group called Public Intellectual Property for Agriculture. I think what I call smart licensing. We don't give a company or anyone exclusive license to everything, like some of the older licenses that went out, like agrobacterium, for example. We license things for the product that the licensee is interested in. So if you're licensed in corn, you get it for corn. If you don't do work in corn, even for 3 years, Davis has a clause that says you need to give it back, and we're going to license somebody else. So we're interested to really get this as broad as possible. And my personal goal is really to get out the developing world. I mean, farmers in the developing world don't use fertilizer, can't afford fertilizer, especially these days, with the conflicts in Russia and Ukraine and things like that. So it's the difference between adding fertilizer or just, having seed that captures some of its nitrogen uses from the air.
So there was a question that came from Julian Alston about wine cultivars, and whether you all have any insights regarding whether gene editing wine cultivars will get to use their traditional names, like Cabernet Sauvignon or something like that, or will they have to lose those? Or do you have any keen insights on that, particularly when those brand names, traditional names, have real value to the consumer?
I don't know how wine grape names are indicated, but I think just, and I think the French may have a lot to say about these things. But I think for fruit trees and like wine grapes, there's different ways of apply gene editing, right? Because a lot of the disease resistance is conferred by a rootstock. So, so the rootstock could be a very different variety than what's grafted on top. So perhaps you never have to change the stuff on top, and you just have to have a more robust rootstock. So I think there is lots of conversations about wine grapes. There's actually, I think Green Minutes, one of our members, is working on wine grapes because they want to decrease the amount of sulfites from wine. So you can decrease the amount of headaches you get when you're drinking. I don't know if that's a good thing or a bad thing. Good thing for me, I think. So I think there's lots of research. It's all what's going to come out and whether there's going to be a pull from the industry, like the winemaking industry.
And I will just say for animals, the breed association. So it kind of, in the case of animals, the breeds own the term Angus or, Red Angus or Wagyu or whatever. They have their own board of directors are making decisions. And in the case of Angus, they have allowed genome edited animals. They're just developing kind of wording to allow those animals into their herd books, and they'll be allowed to use that breed name.
I just want to add, this gets into the space of essentially derived varieties, where you make a small change, usually through back crossing. You might add one trait to, somebody else's variety. So there's certainly laws and rules that regulate that in the seed industry. I think they're pretty well established. But gene editing, I think, will test some of those rules. If I make one change, is it a new variety? Or do I owe the owner at least some sort of royalty or license to do that? Usually the answer is the second one. Right now, the current rules is if you change one trait only, you probably, and that variety is not public, for example, that you've changed, you probably need to be talking to the person who owns that variety.
Yeah, Rick, you want to jump in?
Hey, Jason, I was going to ask if I could go back to your previous question around the consumer traits.
Please do.
Yeah, sorry, I didn't get a chance to jump in earlier on that, but I thought there's maybe something the previous responses were getting at that I just wanted to highlight. So, and by the way, my background, I did a postdoc in yeast, and so having this power to actually go in and quickly manipulate genomes and create edits and modify genes, you can learn a lot. And the field and crops and animals has not had that tool up until recently. So we're still in the stages, and plants and animal genetics has always been behind human genetics anyway. We're just still in this stage where we're learning, and there's a lot to learn by applying these tools. So, I think many of us are interested in bringing, more nutritious fruits and vegetables to market, various different types of consumer traits or sustainability traits. Those are all on, those are on everyone's radar. It's just going to take a little bit of time because, and I'm actually fairly excited because we're starting to see some of those, like the pairwise example, the GABA tomato in Japan, the vitamin D tomato that Kathy Martin's group and others have developed in the UK. So I think it's fairly exciting that we're starting to see those. It's just going to take a little time.
So there's several questions here that kind of relate to the cost of CRISPR type technologies, both the licenses to obtain them. I think, Family, you mentioned there are a variety of different kinds of gene editing technologies. But maybe if anyone would talk about that a little bit, what gene editing can do in terms of cost of creating new plant animals relative to some of the more traditional biotechnologies. And then the other question that kind of came in along there was related to the role of universities versus the public sector or the private sector, and how that cost influences the relationship between those two. So I know there's a lot thrown in there, but anybody want to jump in and tackle some of that? Right, go ahead. You're the brave soul.
Yeah, I'm trying to organize my thoughts, and I think Finley started to address a little bit of it, and agree with what she put in the comments. So a bit of it will end up being, case by case, right? So if you're trying to bring a product to a niche market in a specific geography, costs are likely to be lower than trying to bring something that's a global commodity type of crop, and a lot of the cost and product development starts to scale with the cost of development from a field standpoint. That's where things start getting, that's where R&D really starts to accumulate the costs. So does gene editing make it cheaper? I think, if you think about right now, because of a lot of the regulatory jurisdictions where they're at, and how they're not aligned, as everyone has highlighted, how they're not aligned across the globe, probably not much, because you're sort of forced to go down sort of the GMO type of path that already exists. Hopefully in the future, that cost barrier will start to come down. On the front end, I mean, is gene editing nowadays still, at least in the plant community, still requires a transgenic type of intermediate, or maybe there's other systems, but I think that cost of early discovery is not all that different than many of the other things that we do. I think, as I tried to allude at the end of my talk, I think what editing allows you to do is think about utilizing or doing things differently and scaling the technology differently than has been previously done before with transgenics. That's the key. And then if we can unlock some of those downstream development barriers from a regulatory perspective, I think that's when we'll start to see things moving faster.
Anybody else want to weigh in on that massive a question that I threw out there?
I guess I will.
ahead.
And I'm just speaking as someone at a university, so I'm not commercializing anything, but rather doing research in food animals to maybe test gene function, as has been alluded to. It's a really useful application for that. But this is quite different in terms of all of those animals are unapproved animal drugs, and as a result, therefore, unallowed to enter the food supply, which in the case of cows, that might sound like reasonable enough, but that's not normally what happens with our experimental animals at the university. We have a meat lab, and we sell the meat to the public, whether eggs and milk and all our animal products. And so that really increases the cost dramatically of doing this research when that's the case. And to me, to be frank, when I'm having to incinerate an animal because it's polled, when it's sitting in a pen with other animals that have exactly the same alteration, it's just the height of stupidity, and I get very frustrated by that, and I don't want to spend my valuable grant money burning cows. I'd prefer to support graduate students.
Yeah, I'll echo that. We actually have to burn the plants too, or steam them, but it's not as one plant's a little smaller than a cow, and a little cheaper to burn, or we can burn thousands of plants and probably for pretty cheap, or we actually just steam them. The cost of GMOs has basically precluded pretty much any university and the liability associated with that from every going down that commercial path. We're just not going to take that risk. Yeah, and I know UC Davis just won't do it, and I know most, very few universities will. In gene editing, maybe if that cost changes, we'll take a chance on it. And the cost of development is theoretically of discovery is lower because the efficiency is higher. So we can try more things. Yeah, I think I'll leave it at that. So right now, we're not commercializing anything gene editing. We're partnering with companies who hopefully will co-develop that.
There's a question from Todd Kukin. Apologies if I mispronounced your name, about how the regulatory environment could be improved in y'all's assessment. He references the White House's executive order that asks about ways that we have clarity or efficiency. Any specific suggestions there, or is there an entirely different approach that you think would be more effective for the industry? Bailey, you want to jump in on that one?
Yeah, so I think it's very. I think the regulatory agencies are in a difficult place, especially in the U.S. Is that they're trying to fit a square peg in a round hole, right? So they're trying to kind of take the science and fix it onto regulation, and it doesn't always work out the way they intended. So I think there's a lot could be done in the U.S. framework to have better coordination around the three agencies, understanding that they have different regulatory statutes, different regulatory kind of decision implementations, right? Their sausage making is quite different. But what our industry need, and I think what the research community need, is that the end product is a sausage. We want the end product to be the same. They need to figure out how that occurs, because right now it creates a lot of uncertainty. I think especially for small companies and for university that does not have a bench of regulatory scientists and lawyers to navigate through their processes, right? To read regulation, you need a degree, or you need to have worked in government, right? And that is a barrier to entry for companies. So I think there's a lot that the agencies can do to create a path that is more clear, and that has not happened in the last few rounds of regulatory revision by the agencies. And I think they recognize that by the feedback that the breeding community have given them. So across all the different administrations, regardless of whether stripe they are, this has been a constant to provide clarity, to provide some streamlining for both the animal sector and the plant sector. So these homegrown innovations, like these are American discoveries, can have American benefits. And I think we're not quite seeing that just yet.
There's a very specific question about Bayer's development of short stature corn. Rick, I know you just typed in an answer, but you want to expand on that for the larger group?
Yeah, sure. Thanks. So yeah, we've got 3 paths to short stature corn that are in development towards launch. So one that's an allele that was identified through traditional breeding. That's going to be the first product that we launch. And then we've got a transgenic trait. My background's actually in RNAi, and this one was created with RNAi. So I think this one's actually particularly sophisticated and cool. So the RNAi tunes some specific genes that regulate GA biosynthesis, which then controls height in a very specific way. That one should be launched more towards the end of this decade. And then we have in our early discovery some short stature corn edits that we're trying to work on as well. There was a question about licensing. We try to broadly license we consider broadly licensing all of our products. I would expect that this one should fall into the same bucket.
So Carl Prey asked a question about, there's been a lot created here. He mentions 80 plant varieties, one animal that USDA says isn't regulated, but right now there's not much that's actually been commercialized. And I guess the question is why, from y'all's perspective, what's really keeping some of that commercialization from happening?
I can talk about that a little bit. I think, if you look at USDA's, their not MI regulated, but their confirmation, you see a lot of product development, but I think it's really hard to tell where it is in the pipeline, right? So a lot of things have to happen before a product actually reaches the market. I think Richard and Alan talked about it a little bit. It's like, first, it's proof of concept. You can get it done. And a lot of these things that when they go to the regulatory agencies are still kind of in a research setting, because to have that regulatory certainty is really important before they move on to the next process of product development, because all of these things have to be tested in multiple geographic locations over many years before it can actually come into the market. And it's not just the science, it's not just a regulation. There's got to be a demand, and you have to make sure that the market accepts it. So that's kind of my answer to that. But I think one thing, because we're all US focused folks here, that I kind of want to talk a little bit about and highlight is that, as Allison said, this is a completely different kind of view than the transgenic space. In the transgenic space is a very US, North America focused technology deployment. This is not. This is an international technology deployment. And the consumer attitudes, the consumer benefits are different across the different regions, because those different consumers are thinking about these things very differently. So I think, to focus on the US is a little bit, I guess, not short-sighted, but a little bit myopic, because this technology is going to move in other regions perhaps faster than it will move here. And the consumer attitudes are very different in those countries than it's here, because the consumer pressures are quite different. So I think for me, it's going to be really exciting to see what develops in Japan, in Argentina, in Brazil, and in Africa, because it will I think it would be a very different landscape than the transgenic landscape.
I want to add to that. I briefly said in my talk, I mean, for the first time, the U.S. on GMOs has been a net exporter of technology, in trying to convince other people to buy their products. We need to take it the other side. We're going to get products from Argentina, from Brazil, even from Africa. There's approved bananas in Africa and Kenya that are coming out and things like that. Are we going to, ban those bananas, in from other places? So we're going to become perhaps even a net importer of technology. And so for the first time, the regulators have to think about that. And if, they've never really thought about it at all before.
Maybe one last question. We're getting short on time, but there was a question that was asked by John Bagan about, talking about gene editing technology and what it can do in a precise level. We've spent 20, 30 years trying to convince the public that the traditional biotechnologies are at least no riskier than varieties that are produced through commercial or traditional plant breeding techniques. Do we run any risk of undermining that by elevating gene editing to a different level and suggesting that it's safer perhaps than older technologies? Just curious if y'all have any thoughts on that, about how people perceive these different technologies.
I 100% agree with that comment. I hate to throw the use of exogenous DNA under the bus, if you will, and just be happy with knockouts. I think that's severely limits what we can do. However, having tried to work in this field for my entire career, if there's a possibility that we can get knockouts through, then you can't really blame us for trying. So whilst I think that there's no difference in safety there, just on a product by product basis, the fact that knockouts are not being treated differently to GMOs, and the fact that GMO regulations have precluded the use of the technology, really means that it's no surprise that everything that's been approved to date is a knockout. And that's kind of makes a lot of sense. But I do agree it severely limits, for no logical reason, our ability to introduce useful traits that happen to use DNA that isn't part of the germplasm of that particular species. And as a scientist, that makes no sense. But as a pragmatist, let's get a couple of products out there and get some healthy greens that people like to eat, and let's get some actual food products out there and see what the market does.
Well, great. Well, I'd like to thank all of our panelists for participating this afternoon. I certainly know that I learned a lot and enjoyed the conversation. I also want to thank the Farm Foundation for hosting this conversation as well. And so with that, Sherry, I'm going to turn the mic back over to you.
Great. Thank you, Jayson. And thank you, panelists. I especially enjoyed the last section discussing those global perspectives and the consumer perspectives. I think you've queued up a possible global forum for 2024, given that we often have up to 10 to 20 different countries at our forum. So I appreciate all of your insights today. I'd also like to thank the audience for joining us and submitting your questions. We really appreciate your engagement with us and would love to hear your feedback. So please take a moment to share your comments about today's session in the brief survey that you will receive at the conclusion of this forum. If you would like to help us continue providing value programming such as today's forum, I'll quickly remind you about becoming a friend of Farm Foundation. Check the link provided in the chat to learn more. Each and every gift to Farm Foundation strengthens our ability to address rapidly evolving issues impacting agriculture, the food system, and rural communities. And we are so grateful for your support. Last, I would like to invite you to save the date for the next Farm Foundation Forum, Defining Sustainability, Industry Leaders on Actionable Goals, which will be held on Tuesday, December 12th at 9 a.m. Central. Thank you once again for joining us today and hope to see you next time. Thank you.